My name is Mark Kiel. I'm the Chief Science Officer and founder of Genomenon. Very happy to be here. I've been coming to the show despite my ignorance of the pronunciation for over a decade now. I'm very pleased to be talking about the work that my team has done in better understanding gene-disease relationships using a novel and an actionable approach. This is the agenda that I want to cover. I want to briefly talk about genetic variants, as I think it's very closely in line with what I'm going to be speaking to for the majority of the talk with respect to gene-disease relationships. Speak to the work that Genomenon is doing in our ambition to curate the entire human genome using genomic intelligence. Talk quite a bit about our approach to gene-disease relationship curation. Discuss that novel approach and talk about how those results are becoming actionable. And I haven't seen anyone do this before, but in addition to the agenda, I wanted to lay out some themes. As I was assembling these slides, they struck me, and I want to make sure that they resonate with you as I'm talking. Number one, that you may have experienced, this is an exponentiating problem that we're addressing. And there's an urgent need tied to that problem. To address that problem, there's a scalability challenge, and the way to solve that is through parallelization. So the agenda on the left, for those of you who are more didactic, and the themes for those of you who think more subjectively, both of those sides of the equation are important. As I say, I'll begin with a discussion of genetic variants. So this is a problem that is very familiar to many of you, probably most of you. I have referred to this as the scourge of VUS. This is a problem, and it's compounding over time. It's hard to predict the future, and with the benefit of hindsight, this is perhaps obvious in retrospect, but I would have predicted some years ago that with more and more patients being sequenced, more and more evidence being published and assembled, that the number of variants with undetermined classification would shrink. And in fact, it's proliferating. This is a plot showing the number of VUS relative in ClinVar on the left, and on the right, in time, that count for VUS in gray is clearly growing. And to rise to meet that challenge, as I mentioned, Genomenon is embarking on full curation of the human genome. It's ambitious, and even if we don't get there fully, there will be rewards in striving to meet that ambitious goal. But this is conceptually what a fully curated genome will look like. It comprises every disease and every gene associated with any one of those diseases, and every variant that has been found to be disease-causing in patients who suffer from those diseases, with a review of the evidence in any one of the citing references for those variants and consultation with the relevant databases to render an interpretation. All with that evidence fully annotated and displayed and readily accessible, so that in 10, 20 years' time, this will be a solved problem. As I suggest, that's an ambition. There's that need, and there are problems that we're facing, challenges that we're rising to as we proceed. I mentioned the variants. I'm going to speak a little bit more to variants, and then toward the end, as promised, I'm going to speak about how the problem with variants dovetails with the problem of genes, both of which can sometimes have uncertain significance. So fortunately, there's a framework for understanding how variants are associated with disease. And again, this is very familiar to all of you. It's a structured framework. It relies on evidence from multiple lines, including the clinical circumstances of the patient, but also the population frequency of those different alleles, the predictive scores, but critically and most importantly, the published information from clinical circumstance as well as functional studies. And the interpretations that are rendered span the gamut from benign to pathogenic, with the lion's share of those variants, as we're seeing, being variants of uncertain significance. But not all VUS are created equal. And I quite like this concept of a temperature for VUS. A variant of uncertain significance could be considered to be a variant awaiting final designation as either pathogenic or benign. But there are flavors in addition to categories of VUS. They come from different places. So the temperature is hot VUS, which is more likely to be pathogenic than not, all the way down to, in fact, ice cold, which is abutting on the classification for benign. So I've got a couple of examples of those types of variants, beginning with hot. This is a representative example. They're very easy to find. But a variant that's found in a patient where there's some clinical and even perhaps functional characterization to indicate that though there's insufficient evidence to deem it likely pathogenic, it's probably pathogenic. This is an example from a 2016 paper in the GNB3 disease talking about congenital night blindness. This variant has not been published since, but it was very thoroughly characterized in this paper. It just was insufficient to meet the stated criteria. And on the other side of that equation are the cold variants. This is an example on the right of a plot that's showing variants in the GUS-B gene that are drawn from ClinVar. The red are pathogenic and the blue are VUS. And I'm plotting them against the allele frequency from NOMAD against the number of times those variants may have been cited in patients in the literature. So if you look at the red, you can see a fairly direct correlation between how many patients are found to have that variant in that condition against the carrier status, and that's what you'd expect. When you look at the VUS, unselected VUS now, they hug the x-axis. And if you drop a perpendicular from some of those VUS that have a high carrier frequency, you might predict statistically that they would more likely fall on that x equals y line, but they don't. And when you more closely examine those blue or cold VUS, you find that actually they came from a healthy normal screen. They were found in heterozygous states in individuals not affected by disease. But their designation is equivalent, if you just look at VUS, to the hot variant that I showed earlier. So that's a challenge, and it highlights the need to look very closely at the evidence and scrutinize and examine, but also to think more systematically or holistically about these variants. So that's VUS on either end of the spectrum. Those will probably take care of themselves in time, but I want to talk about the middle of that bell curve. You can call them gray, or I think they're also called tepid. Better said, they're uncertain. They're awaiting designation. And oftentimes, those variants, especially as we're doing more and more exome and genome sequencing, are in genes whose role in disease causation, whose association with disease we're not familiar with. Those could be referred to as genes of uncertain significance, or GUS. The plot that I'm showing here, the colored bars, it's a bit of a red herring. Those are called superstar genes from this paper, a review article about the Human Genome Project. Those are very thoroughly characterized and well known to be associated with disease. Not getting as much play there are the genes in gray, the vast majority of genes that are being published but aren't getting star billing. Those are genes of uncertain significance. They're being published. There's evidence associating them with disease causation. The challenge is there's so many of them. There's a hyperproliferation of that information. That information needs to be harnessed because it has bearing on the accuracy and the appropriateness of our clinical testing. Very similar plot, time and number of citations. You can see an exponentiation in the number of genes that are found to be associated with Mendelian conditions. So this is exponentiating, but nevertheless, there's still many, many thousands of as yet uncharacterized Mendelian conditions. I was just pacing before the talk looking at many of these posters. A lot of those are newly captured in the evidence published in those posters, but there's still a great deal of work to go. This is many thousands of disease characterizations that remain. As many as those that are published, we need to capture those and ensure that they're usable in a timely way. And that results in information overload. Despite the yawning challenge in front of us, there's still too much information for this to be assembled manually. There are very valiant and expert efforts in place to harness this information. We're friends with Clingen. There are many Clingen participants who are Mastermind users. This is work that Sharon Plon and Heidi Ream have published. It's showing in the dark green the pace of gene-disease relationship curations coming out of Clingen. Many of you may be members of Clingen. It's expert work. It's a daunting challenge. It's hard to rise up to this challenge when you're only scaling linearly, when the challenge keeps compounding exponentially year over year. But just as there's a framework for understanding variants of uncertain significance, there's a expert, easy to use framework for understanding GUS. There's classifications for those variants, including disputed, refuted, or as yet no known disease association, very similar to that Bell distribution that I talked about. And this is necessary for us to uncover because, as I alluded to earlier, this is relevant to clinical practice. It's also relevant to drug development, which is top of mind for patients suffering with these disorders. In particular, in clinical practice, it's relevant for variants in that gray arc of the pie chart that I showed earlier that are novel. They have not yet been encountered. As more and more patients are being sequenced, we're encountering more and more of these novel variants. We not only need to better capture the variants that are published, we need to understand the genes that are associated with those diseases and use both of those to better prioritize the sequencing data that's coming out for our patients, increasingly more and more coming out of exomes and genomes. There's detail about that framework. As I said, it's an expert framework. It's one that we hew to closely, looks at genetic evidence at the case level and the cohort level, as well as examination of experimental evidence, looking at functional studies from biochemical to animal models to cell culture models, et cetera. So the challenge that I want to speak to in the latter part of this talk is, can we afford to wait? That linear pace of gene-disease relationship curation that's coming out of Clingen, as qualified and as expert as that is, if you do the math and look at the balance of the genes that have yet to be curated, we would be looking at 25 years to understand all of those Mendelian diseases. That's quite a long time, especially when you reflect on the urgency of the need from a patient's perspective, which I've had the privilege of communicating with a number of those patients before, gives me the fire in my belly to proceed as quickly as possible here. But nevertheless, to do that expertly. So just as Genomenon is curating the genome at the variant level, I'm very proud to announce, as I said, work done by our growing team of expert curators, nearly 100 plus variant scientists. We have curated every one of those genes, comprising the clinical exome, more than 5,500 germline genes. This is in response to our Mastermind, our software search engine. Our users have said, this is important. This is a need that we need help with. And reflecting on our own experience curating the variants in the human genome, the first thing that we would do when embarking on curating the gene at the variant level is curate the gene at the gene level, looking for those disease-gene relationships. And we wanted to present that information to the community, and we're proud to be doing that, announced here at ASHAG through Mastermind. I want to linger on this slide because this is the main impact of the talk here. It's important to emphasize, as I mentioned, that we have a very large team in addition to our internal infrastructure that helps us do this very quickly. This is amounting to 75 curator years of effort. We have done this work in a systematic and scalable way using the gene-first approach. And what I mean by gene-first is to say we took each of those 5,500 genes and we looked for the diseases based on the evidence that indicates that those genes and diseases are related to each other, as opposed to starting with the disease and looking for the genes that are associated with that disease. So that has afforded us a little bit of a broader window into those GDRs, and once we've curated that gene, we have a greater appreciation for all of the diseases that may be associated with that gene. This is a summary of how we've done that. I've alluded to genomic intelligence. There's a couple of leaves on this Venn diagram. One is obviously our focus and passion on and for genetics, but there's also that underlying data component, the computational intelligence that we have brought to bear to understand all the evidence that we've amassed from the scientific and medical literature, that we use to catalog the genetic variants. We also have that information at the gene level. And our curators have that at their fingertips when they're embarking on these gene-disease relationship curations. So it's a balance of the expertise and focus that we have on genetics, the expert people power that we can put behind this scalability, as well as having amassed all of that evidence, which is necessary and vital to accurate interpretation of this information. So the latter slides I have in the form of data and examples. This is one of them. This is a Venn diagram of many of those genes. I say that about 1,500 of the genes aren't reflected on this slide because I put these slides together before they were completed. But what you can see from this representative sample is that there's genes that are shared, genes that are found in the Clingen curations. There are some genes that we've not yet, at least not in this analysis, included that are present in Clingen, but they are curated. But there's also that dark blue crescent of genes, about 2,000. That's not a year, that's actually a number. There's 2,000 genes that aren't present in Clingen presently that our curators have looked at thoroughly, looking for every gene-disease relationship for those 2,000 genes. This is a Sankey plot of how those genes, how those gene-disease relationships for those genes parse. So the question in your mind may be, with what evidence was your curation performed to classify these gene-disease relationships? You might argue that there's 2,000 genes that haven't yet been included in Clingen because they're unimportant. What you can see here is following those guidelines and looking at all that assembled evidence, there's a great number of those 2,000 genes that have very, very definitive, strong, moderate, or even limited, but nevertheless important, gene disease classifications. There's a third of those genes that we've looked at that we've designated as having no association, and it comes in two stripes. One of them, which we haven't yet curated, is there are gene disease relationships, but they're at the somatic level. So we've included that in our curation, but we haven't yet embarked on the somatic aspect of this curation. Many of those 33% of the GDRs from those 2,000 genes fall into that category. And still others have some evidence, but it falls below the threshold for being limited. So to summarize this data slide, it's a very comprehensive reflection of all the evidence for any one of those genes that we've curated that have yet to be curated and published in Clingen or otherwise, showing the strength of the evidence. And here are a couple of examples. These are examples of gene-disease relationships that are unique to Genomenon. It's obviously, we're chasing and cataloging and annotating and scrutinizing the evidence. This is evidence produced by researchers and clinicians like yourselves. This is a gene-disease relationship between ATP2A1 and Brody myopathy. It's from a 2020 publication characterizing 40 patients, including about half who are newly diagnosed and new to this study, and cataloging and characterizing the variance in the clinical circumstance of those patients. So it's a solid, definitive gene-disease relationship. Here's another one, is MPC1 associated with mitochondrial pyruvate carrier deficiency. This is from a 2023 publication. So if you'll recall that exponentiation of the proliferation of these gene-disease relationships being published, this underscores the pace that we're up against, the pace that we need to address in rising to this challenge. It's equally as powerful a story, and the classification here is a definitive disease gene association. The authors demonstrated not only segregation in two separate families and different family members, but also showed functional consequences by studying those variants. So very solid evidence rising to the level of definitiveness. In the middle there are genes that are shared. We've looked at those too, and I'm parsing them now according to whether the gene-disease relationships that we produced and those that are in ClinVar are in agreement or in disagreement. So you'll be happy to know, or as may be expected, there's about 80% perfect agreement. This gene is associated with this disease at the same level, limited, moderate, strong, or definitive. There's 20% that are in disagreement, and again, using that Sankey plot, I want to parse how they disagree and why they disagree. It's not disconcerting disagreement. If you look at the Sankey plot, you can see on the middle category, those are the Clingen classifications, and on the rightmost column, those are the Genomenon classifications. And you can see the lion's share of those that disagree at the bottom are moving upward. They're moving upward in classification. And that is because of the evidence that we've looked at and the recency with which we've curated that information. And I want to show you a couple of those examples before I conclude. So again, these are examples of genes that are associated with diseases that have been curated by Genomenon that have a promotion of their classification from what's published in Clingen. This is one such example. I've got two papers that indicate that there's a designation change. The Clingen classification says limited, and Genomenon's curator's classification says strong, and it's in part based on these studies. This is a 2022 publication demonstrating an association between FLNA and thoracic aortic aneurysm that's familial. And I chose this example not only because of the strength of the evidence in that publication, but also because of the recency of that publication and the fact that the Clingen classification, when last evaluated, was some years ago. So again, this is a challenge. This is a challenging problem. So this is not a slight on the great work that Clingen is doing, mostly done by folks like you. It's to say that this is a challenge. This is a problem, and it's not slowing. It's a sort of breathtaking challenge, the pace of the publications that we need to rise to. And Genomenon is hoping to do its part in solving that challenge. There's another publication that was from a later date, but to emphasize that this evidence is built across multiple publications to rise to these criteria thresholds, in this case promoting the GDR from limited to strong. There's a final one here, going from moderate to definitive, which can have consequences when you're prioritizing variants. If you're in a lab or in a workflow drawing a threshold at strong or above, this might promote that variant if you have a patient where you might otherwise not have suspected this variant in ATP1A1 being associated with Charcot-Marie Tooth syndrome. Again, it's a 2023 publication. So at the crest of that exponentiation of these publications in time that there's no signs of slowing for. So the last thing that I'll comment on here is the actionability aspect of my subtitle, which is to say this information is in Mastermind and it's available to users. We have launched at ASHAG the Mastermind 3.0 interface. It comprises a number of new features that highlight some of these gene-disease relationships that can streamline some of your variant interpretation workflows, but also can be useful for some of those other aspects and applications that I talked about for gene-disease relationships. So this is a screenshot of Mastermind 3.0. Here are some of the component features that I'd like to highlight. There's a gene summary, which promotes efficient curation, and various and sundry curation parameters at the variant level to help speed workflows. But critically, in Mastermind 3.0 are these gene-disease relationships that I've mentioned. These many gene-disease relationships that we're going to be continuing to curate as they're published, comprising 5,500 genes at the germline disease level in the clinical exome, now available to all users of Mastermind. So in closing, I hope I'm doing well for time here. I'd like to connect. We have a contingent of representatives from Genomenon, many of whom have been the driving force behind this work, including Jeffrey Bissonnette, who's our expert director of genomic curation. He's very approachable. I'm actually going to invite him to answer any hard questions I get, if there are any. And then also the mastermind behind Mastermind 3.0 is Brittany Jones. I'm privileged to be working with her. She's our VP of Customer Success and Product Management. And then finally, the woman who has all the answers is Denise Belandres, who is our field application scientist, and she can get connected. She can get you connected to Mastermind 3.0 for demos or training or access. We have a booth at the corner here, 511, and with that, I'm happy to let Jeffrey take any questions. If there aren't any questions, as I said, we're readily available. I'm happy to have conversations with any of you folks about any detail, large or small. Thank you so much. Oh, question.
Thank you so much for this very clear presentation. So I started using Mastermind recently. It was very clear for me to see the pathogenic variants and the benign ones. But how about the variant of unknown significance? I didn't see something that, the way you explained, saying that this VUS is hot or cold. Did I miss something, or it's not actually mentioned in
It's a great question. That's a good thing for me to clarify. The question was in Mastermind, she's a relatively new user. She didn't see the hot and cold designation for the variants of uncertain significance. You're not missing anything, they're not there. It's just a conceptual framework to think about the VUS. It's very finely parsed to say it's hot, it's warm, it's tepid, it's like, cold or ice cold. We don't publish that, but it's, as I said, a mental framework for you to assess the evidence that we've organized in our curations.
Can you give a hint in terms of the gene-disease relationship for risks of disease, which needs like case control studies, and those are usually like, in ClinVar I would say it's fine enriched, but the stat is not well displayed. Perhaps how you present those data on your platform.
Yeah, that would be a detailed question for Jeffrey. The question was about disease risk as opposed to definitive disease gene causation. For those that aren't, in the lion's share of the types of diseases, I would invite you to ask Jeffrey. But as I say, anybody else who has any other questions or would like to see Mastermind 3.0 and some of these gene-disease relationships, I invite you to come to the booth. Thank you so much.